In this demo, we're going to investigate the effect of an environmental factor on species distribution. In the previous video, we learned how to perform random sampling. What about systematic sampling? Well, as we mentioned previously, in systematic sampling, samples are taken at regular intervals. An example of a systematic sampling technique is the interrupted belt transect. So let's write a hypothesis and learn how systematic sampling can be performed. Let's say we decided to investigate the effect of light intensity on the distribution of daisies on a lawn. And suppose that this is our lawn. Here we have a tree, and the right circles are daisies. As you can see, this area is shaded because of the tree, and as we move, light intensity increases. Then, what would be the hypothesis? Keep in mind that a hypothesis is a precise, testable statement of what the researcher predicts will be the outcome of the study. Then, the hypothesis is that there is a correlation between light intensity and the distribution of daisies on a lawn, meaning that we're predicting that there's a relationship between light intensity and the distribution of daisies on a lawn. Now for the sampling method, remember, we said previously that the systematic sampling is used to investigate a correlation between a factor and an organism. So obviously we should use the systematic sampling technique because we are investigating how daisies differ throughout the length of a lawn, from the shaded area under the tree to the sunny area away from the tree. So there's an environmental gradient, meaning as we move away from the tree, light intensity increases. And of course, we should choose a large sample size, which is suitable for statistical analysis. So a minimum of 10-meter tape measure is required in order to acquire data from at least 10 quadrats. This will be discussed later on during investigation. Now, the equipment needed for this method are a quadrat, a tape measure, a pen, and a paper. Now let's start. Step 1. Extend a measuring tape from the tree to the end. So the area around the tree is the shaded area, and as we move, light intensity increases. Step 2. Place the quadrat at 0 meters on the tape. Step 3. Count the number of daisies inside the quadrat, as we used to do in random sampling, and record the data in a suitable table. Similarly, we can estimate the percentage cover of the daisies. And step 4. Move the quadrat at regular intervals and count the number of daisies each time. Note that you should keep doing this until the full length of the measuring tape has been sampled. Now, to be more scientific, you also need a method to measure the factor you're investigating. So here we are investigating light intensity. So we can use a light meter and measure the light intensity every 1 meter as we move the quadrats.